notes on meiosis. I'm going to say meiosis. Sometimes it's pronounced meiosis. I'm just going to say meiosis so we can differentiate between mitosis and meiosis. Okay?
Okay.
All right. So we're going over meiosis today. This is how you get
Me.
yes, how you get me. How you get a person. So these are how sex cells are developed. So, some terms that we're going to go over first that you do need to know. Gamete is just a broad term for sex cells that could be sperm or egg. The egg, or sometimes it's going to be called ovum, is the female sex cell. Sperm is the male sex cell. And then when the sperm fertilizes the egg, it becomes a zygote. So this is a fertilized egg. You did start that video, didn't you?
Yeah.
Okay. All right. Spermatogenesis is just how sperm is made. What do you think oogenesis is?
How over?
Oh, in the ovaries are the
Eggs.
eggs. So this is how eggs are made. And then you're going to hear me referring to homologous chromosomes. These are just going to be the chromosomes that are passed along in sexual reproduction. So when the zygote becomes fertilized, there are chromosomes that are passed along. And when I'm talking about meiosis, I'm going to be referring to homologous chromosomes, but that's just the chromosomes that are being passed along. You have the first ones, right?
Yep.
Okay.
Sure.
Does it ever have sperm and zygote?
Yep.
Yeah.
Why
Okay, so meiosis goes through basically 2 stages, just like we've already talked about with mitosis. So it's going to have the PMAT part of it times 2. So what does the P stand for? Prophase.
Yep. Phase.
So there's prophase 1 with it. Okay, this is where the nucleus breaks down. And the chromosomes condense. This is also where something called crossover happens. Okay, so crossover is a newer term that we've not really learned about. So if you have 2 chromosomes, and you know that chromosomes are kind of little X-shaped looking structures. With crossover, it's literally crossing over and touching the other one. So see how they're crossing over right there? But what they're going to do is they're going to exchange information. So after they cross over, this this one will now have a little section of the green one in it. And then the green one, where it touched the red one, will have a little section of the red in it. So it's just exchanged genetic information. You will probably see a test question on your test that looks something like this. So it'll show these two chromosomes crossing over, and then it'll show you a picture like that, and it'll ask you like what's going on or something along those lines. So that's just crossover or an exchange of genetic information. Okay, so if we have a little diagram of prophase 1, it's just kind of got little chromosomes in it. There's the nucleus is starting to break down. You may see a little bit of it. All right, so that's prophase 1. What's the next phase going to be?
Metaphase.
Metaphase. So it'll be metaphase 1. Same thing as before. The chromosomes in metaphase line up down the middle. Let me do this. They actually do 2 rows. That's better. Alright, so with metaphase, the chromosomes are pairing up. And line up in the middle. Does it ever have the prophase 1?
Yep.
I'm trying not to write down low because I know some of you can't see that. Alright, so then we've got anaphase 1. Okay, and remember anaphase is where this stuff gets pulled to the sides. Okay, so we're pulling the chromosomes to the sides. Alright, so we're going to move the little chromosomes over to the sides. And those are pulled by spindles. And then the last phase is what? Telophase. And this is telophase 1 because with meiosis it goes through this process twice. So telophase 1, the chromosomes are going to gather on the poles of the cell and the cytoplasm divides. When it actually divides, what is that?
Cytokinesis.
Yeah, cytokinesis. So this would be the chromosomes. The nucleus is going to start trying to reform. And then it's going to start trying to break up down the center. Alright, so we went through the PMAT thing once. Now we're going to go back through it again. So it just starts all over with the first phase, which is prophase, except this time it'll be prophase 2. So the fifth step is prophase 2. So you've got your cell, well, you've got both of the cells that it just made. So we're starting with these 2 cells. Okay, so we've got our nucleus that's kind of breaking down. It's got some chromosomes in it. And we got the little spindles that are starting to form in it. And then we're going to go to metaphase 2. So these chromosomes that are in it are lining up in the center. And then you're going to have anaphase 2. What's going to happen with anaphase? You've got these little spindles that are going to be pulling these chromosomes, but in anaphase 2, it pulls the chromosomes in half. Okay? So it's going to pull half of them over on this side and half of them over on this side. So now we'll have like a half of a chromosome over here, and then we'll have the other half over here. Alright, so we're going to pull the chromosomes in half. And the little, what it's actually doing, the chromosomes have like this little thing that kind of holds the sides together called a centromere, and the centromere is actually just kind of breaking down, and then the spindles are pulling it over there. But just generalize, you're just pulling the chromosomes in half. And now, since it's not a full chromosome, it's something called a chromatid, which is just an area that has genetic information, area on a chromosome. And then the last phase is telophase 2. So this is going to be where the nucleus develops around the genetic info, and the cells split. So if these cells are splitting, what are they splitting into? How many?
2, 4.
We've got 2 that are splitting, so in the end, so you've got one cell that's going to split with its stuff in it, and then you've got another cell that's going to split with its stuff in it. So in the end, we should have how many cells?
2.
No.
No.
4. So the end result is 4 cells that are haploid cells. Haploid means they have half the genetic info. Why do you need half of the genetic info?
It's half from
Because it's
each
half
parent.
and half between the two people.
Yeah, you can't have somebody donate all their genetic info, like the mom and the dad, and get a baby out of it. Mom has to donate half of her genetic info. The dad has to donate half of his. And then, so they're donating 23 chromosomes, each one of them, to make 46 total. So after it's fertilized, what kind of cell would it become? It's not haploid, it would be the one that has 2 sets of DNA in it, or all the sets. Diploid. So a zygote, remember that is a fertilized egg, will be a diploid cell. Now, sometimes these are referred to as daughter cells. So if you see that, that's just what it's talking about. It may ask you how many daughter cells are produced in mitosis. It may ask you how many daughter cells are produced in meiosis. Mitosis produces how many cells? 2. Meiosis produces 4. Mitosis, does it have all the genetic information or half? Mitosis.
Mitosis.
Has all of it.
Have all of it, and
And then meiosis has half. Okay, so things that are important with this, and these will be things that you'll probably see on your test, but you'll be asked about how sexual reproduction has more advantages than asexual reproduction, something along those lines. So this has something called independent assortment. So I'm talking about sexual reproduction here. So with the crossover, wherever those chromosomes touch, it exchanges genetic information. They don't always touch in the same place, so they can have an independent assortment, a variety of areas that they touch and genetic information that's exchanged. So sexual reproduction has independent assortment, so it's just a variety. Choice is not a really good word there. Choices or genetic info. So if you look at a family, like a bunch of brothers and sisters, they may have similar characteristics, but for the most part, they don't all look identical because there is independent assortment. There is the crossover. This is because of crossover.
How does twins work?
Well, twins can work different ways. A lot of times what happens is if they're identical twins, the egg that's fertilized will start to grow, and then all of a sudden it will split into 2. So that's why it's identical genetic information. There are twins that are not identical, that are just 2 eggs came down in the fallopian tubes and got fertilized. But yeah, that's the gist of it. And let's see, sometimes you hear them talk about random fertilization. What that's talking about is just the woman's ovaries can produce multiple eggs. Most of the time it's just one. They can produce multiple eggs. So whatever egg comes out of the ovaries and into the fallopian tube, that's got that genetic information in it. The next time that egg might not have the same genetic information in it. And whatever sperm that fertilizes the egg is kind of random too. So you don't know which sperm is, and all the sperm are carrying different genetic information. So it's not like you're going to get the same baby. Even if you have two eggs that are fertilized at the same time, those could be twins, but they're not necessarily going to look the same because this sperm fertilized this one and this sperm fertilized that one, and they're not necessarily carrying the same genetic information. Identical twins are different because that's where the fertilized egg splits. So it splits the genetic information that's already in there. Okay, so asexual reproduction doesn't have an assortment. No assortment. It doesn't have a variety to it. It's just an exact copy. And examples of asexual reproduction are binary fission. So this is exactly what it says. What is bi mean?
1 or 2.
2. Fission means to do what?
You divide.
Yeah, so it's going to split or divide. So this would be like a cell that you have, and it splits into 2. And now you have this cell and that cell. They have the same genetic information. Or it could be something called budding. Sponges do this a lot. I don't know if you've ever seen pictures of sponges, but like a little sponge will be growing in the ocean, and then it'll get like a little branch, it looks like, off of it. And then that little branch will eventually pinch off, and it'll settle down here, and it'll start to grow into another sponge. But it is an exact copy of it. Okay, so it's not.
So potatoes.
Yeah.
If you never eat them.
Huh?
If you never eat potatoes and you grab a potato and you see like a whole nother potato.
Can I bring a coral in tomorrow?
Yes, you can bring your coral in tomorrow.
Thank you.
Okay, so what you're going to do right now is you're going to, and I've got the assignment in Canvas, but you're going to work on a meiosis diagram. So there's white paper up here, there's color pencils, there's markers and stuff up here too. But I want you to look at the directions, because I told you to label where crossover happens and a few different things happen. But I want you to diagram out the stages of meiosis. That's meiosis 1 and meiosis 2. Show the cells, tell what they're doing. Okay.
Kind of dry it.